Evolutionary tree of life. DNA analysis is showing how we got so much wrong. If you look different from your close relatives, you may have felt separate from your family. When you were younger, you could have even believed that particular tumultuous fallouts were a sign that you were adopted. According to recent studies, appearances can be deceptive when it comes to family. The family tree of many plants and animals is being upended by new DNA technology. Before we go further, what is DNA? DNA, deoxyribonucleic acid, is a molecule that holds the biological instructions that gives its species its individuality. During reproduction, DNA and the instruction it carries are transferred from adult organisms to their progeny. The instructions required for a creature to grow, survive, and reproduce are encoded in its DNA. To carry out these functions, DNA sequences must be converted into messages that can be used to produce proteins, which are the complex molecules that do most of the work in our bodies. According to recent research conducted by scientists at the Milner Center for Evolution at the University of Bath, inferring an organism's evolutionary history from comparisons of its anatomical features rather than its gene sequences may be deceptive. The study, which was published in Communications Biology, shows that we often need to overturn centuries of scholarly work that classified living things according to how they look. Due to significant physical similarities between our skeletons and brains, primates of which humans are a member, were traditionally assumed to be closely related to bats. However, DNA evidence now classifies humans with other animals like rodents, such as rats and mice. Surprisingly, it turns out that bats have more in common with cows, horses, and rhinoceros than with humans. Scientists in Darwin's time and through most of the 20th century could only work out the branches of the evolutionary tree of life by looking at the structure and appearance of animals and plants. Life forms were grouped according to similarities thought to have evolved together. About 30 years ago, scientists began creating molecular trees using DNA data. Many of the first trees based on DNA data were at odds with the classical ones. Sloths and anteaters, armadillos, pangolins, and aardvarks were once thought to belong together in a group called edentates, since they share aspects of their anatomy. However, with the development of quick genetic sequencing methods, biologists are now able to use genetic data to piece together evolutionary relationships for species very quickly and affordably, frequently demonstrating that organisms we previously believed to be closely related belong in completely different branches of the tree. For the first time, scientists at Bath compared evolutionary trees based on morphology with those based on molecular data and mapped them according to geographical location. They discovered that animals grouped using molecular trees live geographically closer together than animals grouped using morphological trees. Matthew Wills, professor of evolutionary paleobiology at the Milner Center for Evolution at the University of Bath, said, "It turns out that we've got lots of our evolutionary trees wrong. For over 100 years, we've been classifying organisms according to how they look and are put together anatomically, but molecular data often tells us a rather different story. Our study proves statistically that if you build an evolutionary tree of animals based on their molecular data, it often fits much better with their geographical distribution. Where things live, their biogeography, is an important source of evolutionary evidence that was familiar to Darwin and his contemporaries. For example, tiny elephant shrews, aardvarks, elephants, golden moles, and swimming manatees have all come from the same big branch of mammal evolution, even though they look completely different from one another. Molecular trees showed that these traits evolved independently in different branches of the mammal tree. It turns out that aardvarks are more closely related to elephants, while pangolins are more closely related to cats and dogs. Darwin and his contemporaries were aware of another significant line of evidence. Darwin observed that organisms with the most apparent shared ancestry were frequently found geographically close to one another. Another major signal that two species are linked is their geographical proximity. Species that coexist close have a higher likelihood of having a common ancestor. The team at the University of Bath looked at evolutionary trees based on appearance or molecules for 48 groups of animals and plants, including bats, dogs, monkeys, lizards, and pine trees. Compared to conventional evolution maps, evolutionary trees based on DNA data were 2/3 more likely to match the location of the species. To put it another way, older trees demonstrated how various species were connected based on appearance. In contrast to species connected by DNA, research revealed that they were much less likely to coexist in close proximity. It may appear that evolution endlessly invents new solutions almost without limits, but it has fewer tricks up its sleeves than you might think. Animals can look amazingly alike because they have evolved to do a similar job or live in a similar way. Birds, bats, and the extinct pterosaurs have or had bony wings for flying, but their ancestors all had front legs for walking on the ground instead. Because the physics of producing thrust and lift in the air are always the same, similar wing forms and muscles evolved in many groups. The evolution of eyes, which may have occurred 40 times in animal evolution and have only undergone a few simple designs, is very similar. Our eyes, which have crystalline lenses, iris, retinas, and vision pigments, are comparable to squids' eyes. Squid are not as closely connected to us as we are to snails, slugs, and clams. However, many of their molluscan relatives have quite basic eyes. At least 4 times on several continents and branches of the mammal family tree, moles developed into their current form as blind, burrowing creatures. The African golden mole, the Eurasian and North American talpid moles, and the Australian marsupial pouched moles all evolved along a similar evolutionary path. Professor Willis said, "We already have lots of famous examples of convergent evolution, such as flight evolving separately in birds, bats, and insects, or complex camera eye evolving separately in squid and humans. But now with molecular data, we can see that convergent evolution happens all the time. Things we thought were closely related often turn out to be far apart on the tree of life." People who make a living as lookalikes aren't usually related to the celebrity they're impersonating, and individuals within the same family don't often look alike. It's the same with evolutionary trees, too. It proves that evolution just keeps on reinventing things, coming up with a similar solution each time the problem is encountered in a different branch of the evolutionary tree. It means that convergent evolution has been fooling us, even the cleverest evolutionary biologists and anatomists for over 100 years. Dr. Jack Oyston, research associate and first author of the paper, said, "The idea that biogeography can reflect evolutionary history was a large part of what prompted Darwin to develop his theory of evolution through natural selection. So it's pretty surprising that it hadn't really been considered directly as a way of testing the accuracy of evolutionary trees in this way before now." What's most exciting is that we find strong statistical proof of molecular trees fitting better not just in groups like Afrotheria, but across the tree of life in birds, reptiles, insects, and plants too. It being such a widespread pattern makes it much more potentially useful as a general test of different evolutionary trees, but it also shows just how pervasive convergent evolution has been when it comes to misleading us. Up until the development of affordable and effective gene sequencing technologies in the 21st century, evolutionary biologists frequently had little other information to go on than appearance. Darwin, 1859, demonstrated the interconnectedness of all life on Earth in a single evolutionary tree, although he did little to depict the tree's branches. One of the first individuals to depict evolutionary trees that attempted to illustrate the relationships between significant groups of life forms was the anatomist Ernst Haeckel, 1834 to 1919. Ernst Haeckel was a German zoologist, naturalist, marine biologist, physician, and artist. He discovered, described, and named thousands of new species, mapped a genealogical tree relating all life forms, and connected many terms in biology, including ecology, phylum, phylogeny, and protista. Haeckel's drawings made brilliant observations of living things that influenced art and design in the 19th and 20th centuries. His family trees were based almost entirely on how those organisms looked and developed as embryos. Many of his theories regarding the relationships between species were accepted until recently. There will be more surprises as it becomes cheaper and simpler to gather and analyze huge amounts of molecular data. Evolutionary biologists are always debating and improving the phylogenetic classifications. Every year, fresh information appears that modifies phylogenetic tree diagram linkages in new ways. Prior to the development of molecular biology, phylogenetic trees were built based on homologous and analogous morphology. Nowadays, however, molecular analysis data is used more and more frequently. Many evolutionary relationships in the modern tree have only recently been determined due to molecular evidence. Nucleic acid and protein analysis have informed the construction of the modern phylogenetic animal tree. These data come from a variety of molecular sources such as mitochondrial DNA, nuclear DNA, ribosomal DNA, and certain molecular proteins. Evolutionary trees can be made by the determination of sequence information of similar genes in different organisms. Sequences that are similar to each other frequently are considered to have less time to diverge, while less similar sequences have more evolutionary time to diverge. Sequences are aligned and each branch's length are proportional to the sequence's variations in amino acids to produce the evolutionary tree. Furthermore, by assigning a constant mutation rate to a sequence and performing a sequence alignment, it is possible to calculate the approximate time when the sequence of interest diverged into monophyletic groups. A change to former phylogenetic groupings because of molecular analysis includes the emergence of an entirely new phylum of worm called Acylomorpha. These acyl flatworms were long thought to belong to the phylum Platyhelminths because of their similar flatworm morphology. However, molecular analysis revealed this to be a false relationship and originally suggested that acyls represented living species of some of the earliest divergent bilaterans. Well, as time goes on, more discoveries will be made. That's the end of today's video. Thanks for watching. What do you think about this discovery? Please drop your comments in the comments section below. Also, ensure you like, share, and subscribe to our channel to get more amazing and enlightening videos. You can also click on the bell icon to turn on post notifications so you can get alerts anytime we upload a video. Bye for now, and see you in the next video.